So, hello students, welcome back to this lecture series on animal cell culture technology. I am Dr. Joyjyoti, Assistant Professor of Biotechnology at Amas University. So, in this lecture, we are going to discuss about cell culture media, which is a very important topic in the context of cell culture because uh, when you're culturing cells in the laboratory, you are definitely going to need uh, to provide food to the cells. And that is why we have kept it as a separate uh, lecture topic in this series, because it is very important and it needs to be discussed at length uh, in our uh, in our video lectures. And uh, along with it, we are going to discuss the various types of contamination that may occur while handling animal cells, and therefore they need to be avoided. So unless we are aware of how or what contamination might occur in cell culture, we won't be able to avoid it. So if you remember, if you recollect from the last lecture, we talked about the design of an animal cell culture laboratory, the various essential elements that need to be present in an animal cell culture laboratory. And we also discussed the basic equipment that are required for culture of animal cells. In this lecture, we are going to talk about the food for the cells, which is called as the cell culture media. So let's start with the definition of growth media. So from microbiology, we all know that a cell culture medium is a single most important factor in promoting cell survival and proliferation. Uh, Let it be bacterial cells or let it be animal cells or plant cells. If you're growing the cells uh, in a controlled conditions in your laboratory, you have to provide them with nutrition that matches with that of the host the cell belongs to. And so medium is going to play a very crucial role. A nutrient is defined as a chemical substance that enters a cell and is used as a substrate for biosynthesis or energy metabolism. So we all know that for any living cell, these are two very important activities that are always going on inside the cell. One is the synthesis of the biomacromolecules, which are nucleic acids, proteins, lipids, and also at the same time, the uh, metabolism of various metabolic substrates to produce energy in the form of ATP. So all that will come from nutrients that are being provided to the cells. Any other requirement for cell growth is termed as a supplement. And that includes, in case of animal cell culture, what we call serum. It also includes undefined media and other biological fluids that may be of nutritional importance to the cells of interest. The function of media is to provide an environment for survival and also to provide substances required for the growth of cells. So survival is not only enough. So you have to provide nutrition that will aid the growth of the cells. Only then they will divide and produce more cells of their kind. Early tissue culture was done on biological fluids such as plasma, lymph, and serum or extracts of embryonic origin, right? So, just to revise, serum is that component of the blood which gets separated when the blood cells have uh, coagulated and settled at the base of the tube. So the yellow color fluid that stays above is the serum. While if while during the process of blood cell separation, you're using an anticoagulant and you're not letting the fibrinogen and other substances form a clot, then what you get in the yellow color fluid is the plasma, right? So uh, these body substances or body fluids rather were used primitively for culturing cells as they carry a lot of nutrition in them and they are enough to sustain the growth of cells in culture. So besides meeting the basic nutritional requirement of cells, the cultured medium should also have any necessary growth factors, regulate the pH and osmolality, and provide essential gases, oxygen and carbon dioxide as and when need be. So now let's categorize media. So we talked about biological fluids, and we said that they were initially used in cell culture media. But then you can well understand again, if you need to extract biological fluid, you also need to get some organisms from which you can extract them. And of course, again, the ethical concerns will be coming into considerations because you might be needing to sacrifice the animal or cause pain to the animal to extract that biological fluid from that particular animal. So, though natural media was primitively used, but then scientists started coming up with chemically defined media or media composed of inorganic and organic compounds. Okay, so let's talk about the first category of media, which is natural media. What constitutes natural media? It consists of natural biological substances such as plasma, serum, and even extracts of embryo, which are highly rich in nutrition. Okay, so coagulants or clots, for example, can be used as natural media for cell culture. Tissue extracts, extracts of chicken embryos, liver, spleen, and bone marrow extract have been used for cell culture, along with the biological fluids that we already talked about, plasma, serum, lymph, and even amniotic fluid or pleural fluid found in bodies of animals. So uh, next we come to the other category, which is the synthetic media. So media that is not occurring naturally, instead being synthesized. That is, of course, what we refer to as synthetic media. And it is composed of a basal medium. And along with that, there will be supplements such as serum, growth factors, and hormones to support the growth of cells. So under synthetic media, media can further be categorized into serum-containing media, serum-free media, and xeno-free media. So serum-containing media will contain the serum component, of course, as a supplement, and the serum-free media will not contain serum, but it will contain crude protein fractions such as bovine serum albumin, alpha or beta globulin, etc. And xeno-free media means the supplements will be from human source. Okay, and there will not be any other animal components supplemented in xeno-free media. So there are two other categories of synthetic media or artificial media, as we may call it. So one will be called as protein-free media, wherein there will be undefined components such as peptide fractions. There will not be whole proteins there, but protein hydrolysates to supply amino acids to the system. And the other category, which is the most popular category of cell culture media, is the chemically defined media. Because here you are not using anything which is of undefined composition. You're not using undefined components such as crude protein fraction, hydrolysates, or tissue extract. So instead, you're actually supplementing the media with highly purified components such as recombinant proteins and other appropriate supplements. And the inorganic part of the media is completely well measured and calculated and chemically defined. Okay. With that knowledge, we go to our next slide, wherein we'll be further breaking down the term basal medium. Okay, so we said a basal. What is a basal medium? We talked about basal medium in artificial media. So basal media is an unsupplemented medium which promotes the growth of many types of microorganisms and even uh, animal cells and do not require any special nutrient supplements. Okay, so this definition holds true for basal media in case of animal cell culture as well. What you see in this figure here is a basal media that is manufactured by a company, cell culture based company called Lonza, and this is available for ready use in your laboratory. Whereas the other form of media that comes from the company is in the form of a powder, and you need to dissolve this powder in autoclaved water to make up your final media and add supplements along with it to apply to your cell culture. So now let's talk about balanced salt solutions. So balanced salt solution, this is something that is composed of inorganic salts that maintain the physiological pH and osmotic pressure. The physiological role played by the inorganic ions is to maintain the membrane potential. We all know the utility of various ions in animal systems, right? Like to maintain the osmolality, concentration of sodium chloride is adjusted. Four main categories of BSS are that are available are EBSS, HBSS, DPBSS, and ESSS. All have all are acronyms or short forms of a big name associated with the balanced solution. So why does a basal salt solution serve the purpose of keeping your cells alive for some time? Because uh, it will maintain the osmotic pressure of the cell, and it will also provide ions which are required uh, as cofactors in various enzymatic reactions and cellular attachment, right? So if you provide balanced salt solutions to your cells, they can survive and also grow in them, but not for long-term cultures. You have to supplement them with other constituents for long-term culture. Okay, a very commonly used balanced salt solution in routine cell culture laboratories is the phosphate buffered saline, the what we call as PBS, which comprises sodium chloride, potassium chloride, sodium bihydrogen phosphate, and disodium hydrogen phosphate in autoclaved water. And the pH is maintained around 7.4, which is the physiological pH. Okay, so let us discuss more on the basic components of cell culture media, and let us try to understand why these need to be present in animal cell culture media. What is the role of the specific component in animal cell culture media? So, what are the basic components? Amino acids, sugars, inorganic salts, vitamins, a natural buffering system, phenol red, proteins and peptides, and trace elements. Let's talk about these. So, uh, these are texts. These are excerpts taken from the book of Culture of Animal Cells by R. Young Freshney, and wherein the role of the individual components are discussed. And we know that carbohydrates are the major source of energy in cultured cells. So sugars such as glucose, maltose, sucrose, mannose, fructose, and galactose are commonly used. Glutamine can also provide alternatively energy to some cells. Animal cell culture requires essential amino acids as they cannot be synthesized from raw materials by heterotrophic organisms. So you need to provide amino acids to your cell culture system. Okay, and why are vitamins required? Vitamins are required as cofactors in metabolism, uh, and we all know that vitamins added to basal media. Some examples of that are para-aminobenzoic acid, biotin, choline, folic acid, nicotinic acid, pantothenic acid, etc. Sufficient supply of choline is necessary for incorporation into membrane phospholipids because we know that choline is a very important part of the phospholipid bilayer. Okay, and so all these need to be supplied to the cells externally. Why do we need antibiotics? Okay, even in bacterial cell culture, we sometimes use antibiotics. But why is it required in case of animal cells? Of course, to prevent the growth of microorganisms in your animal cell culture media, because you want your animal cells to completely consume the media and not share it with bacterial cells or any other microorganisms, because once they start growing, they will contaminate your cell culture completely and your cells, animal cells, will die. So, basic dose of antibiotics is supplied to the media, and the commonly used antibiotics are penicillin and streptomycin. Gentamicin and nystatin are also used as antifungal agents at times. So, a very important uh, point of differentiation with uh, uh, microbial cell culture is that uh, we do not need carbon dioxide supply for our microbial cells, but for animal cell, carbon dioxide is a necessity. Why? So animal cell culture requires a gas phase consisting of oxygen and carbon dioxide, although the oxygen requirement by cells and organs may differ. Uh, Carbon dioxide that you supply to the system through the carbon dioxide cylinder that we saw in one of our previous lectures attached to the incubator, it does maintain the pH of the system by serving as a bicarbonate buffering system. Okay, so uh, you have to constantly keep your media supplied with enough carbon dioxide so that the buffering of the buffer range of the media stays around 7.4, which is again the physiological pH. Okay. Eh, when cells are growing, they are uh, uh, obviously respiring, and they are also producing metabolites. Various metabolites are which are making the pH of the culture acidic in nature. Okay, so if CO2 is not added to the media, then which obviously contains some other chemicals that can serve as a buffering system. In this case, sodium bicarbonate is used. So unless CO2 is added, the buffer of the system will not be maintained to a pH of 7.4. So that is why it's written here, media needs to be buffered to compensate for the evolution of carbon dioxide and the production of lactic acid from glucose metabolism. Bicarbonates form a cheap and most abundantly used buffering system. But in certain cases, you can avoid the use of exogenous carbon dioxide gas by instead using HEPAS. So HEPAS stands for N2-hydroxyethylpiperazine N2-ethane sulfonic acid, and it has been used to maintain the pH of 7.4 in cell culture media. So what do you use as a supplement? You, of course, in modern day culture, you use supplements, and what you use here is serum. Okay, so as I said, serum is plasma minus fibrinogen, and it may be used either as an entire culture media or as a supplement in basal medium. So most often or most frequently, we use serum as a supplement in basal media. And serum provides a nutritive substrate and a supporting structure of many types of cell culture. Okay, most importantly, it contains attachment factors, which helps the growth of adherent cells. And we have discussed this before. Okay. So now serum is a biological fluid and it has uh, several constituents. It has growth factors, which are important for cell division. It has trace elements, which are all important uh, elements in cell systems. It has lipids, steroids, attachment factors, which is the most important, and it also has some proteins which help in transport of ions. And finally, serum also contains protease inhibitors, which is important for neutralization of trypsin. We have already discussed uh, the process of subculturing, wherein we have used trypsin to dislodge the cells from the substrate. So, because there are protease inhibitors in serum, so after neutralization, after lifting of the cells with trypsin, we use the complete media which contains serum to neutralize the activity of trypsin. And so there are growth factors also in serum and protein. And what are these? These are proteins of molecular weight, 5 to 30 kilodalton. Uh, and we have proteins such as albumin, as we saw in the last table. It's a major protein component of serum. It acts as a carrier for small molecules across cell membrane. Transport of fatty acids is an important function of albumin. Steroids and fat-soluble vitamins also bind to albumin. And lipids such as cholesterol, triglycerides, and phospholipids are transported in serum in micellar form, complex with specific lipoproteins. So it also acts as a pH buffer, protecting the cells against damage. What is the role of transferrin in serum? It is a major iron transport protein in vertebrates. Apart from that, there are attachment factors and that serum provide, and name of those attachment factors are fibronectin, ferritin, and laminin. This we discussed while discussing about cell adhesion. And as I said, there are protease inhibitors also in serum, examples of which are aproteinin and alpha-2 macroglobulin. And so this table gives you a summary of the various types of components present in serum and their respective function. So now let's compare the advantages of serum in media and why you would not prefer having serum in media. So serum contains various growth factors and hormones which stimulate cell growth and functions. That is an important advantage, but there's lack of uniformity in composition of serum because you're directly isolating a biological component from a microorganism, from an organism, and it may vary from batch to batch. While serum helps in attachment of cells, testing needs to be done to maintain the quality of each batch before using. Otherwise, there might be variations in your experimental data. Serum acts as a spreading factor. It acts as a buffering agent, which helps in maintaining pH of culture media. It functions as a binding protein, minimizes mechanical damages or damages caused by viscosity. But then it may contain some of the growth inhibiting factors as well, and it may increase the risk of contamination because it's a highly nutritive source and it may attract uh, microorganisms. Okay, so with the addition of serum, the contamination chances of your cell culture media keep on increasing. And at times, presence of serum in media may interfere with the purification and isolation of cell culture products if you are trying to extract and isolate some components secreted extracellular by the cell in the media. Okay, so with that, we'll come to the next part of our lecture, which deals with the sterilization of culture media. And in my previous lecture, I have discussed about the various techniques of sterilization of objects that you use in cell culture. And I said that cell culture media is not autoclaved, and there is a reason why it is not autoclaved, instead filtered, because now we know it. Now we can like now we can easily comprehend why, because there are proteins and peptide fractions present in cell culture media which are heat labile, and so they cannot withstand temperature of 121 degrees Celsius. So what needs to be done instead is membrane filtration, wherein you use a filter and pass your medium through that filter. That filter will be having a range of zero point 0.2 microns, say, which prevents the passage of any contaminating microorganisms through it. And so what you receive down in this reservoir is your filtered media. So this is the realistic view of the filter that you use or the setup that you use for membrane filtration. And to create a negative suction, what you attach to the nozzle of the bottle in which you are collecting the media is a vacuum pump. So this creates a negative pressure, and the media then descends down through the filter and gets filtered. Alternatively, for small volumes, you may use such sterile filters, membrane filters, or syringe filters in this case, wherein you can attach the filter to the mouth of a disposable syringe, and through that you can pass the media or antibiotic or any other solution that you are going to use in cell culture and get the solution filtered. So the nylon membranes present in such filters are generally of 0.22 or 0.45 micron pore sizes. So now we come to the last part of our lecture, wherein we are going to discuss about the common cell culture contamination. They may be of two types, either chemical contamination or biological contamination. So chemical contamination or chemical contaminants such as impurities in media, sera, and water, endotoxins, plasticizers, and detergents, all such materials can serve as chemical contaminants in your media, and you need to be very careful while handling your media. And biological contaminants may include various types of microorganisms such as bacteria, mold yeast, viruses, mycoplasma, as well as contamination by cross-contamination by other cell lines when you are performing uh, experiments related to co-culturing of cells, when you're handling several cells together. So that can be also a major source of contamination. So while not as common as microbial contamination, extensive cross-contamination of many cell lines with HeLa and other fast-growing cell lines is a clearly established problem with serious consequences. So if there is a contamination, what you need to do, you need to discard your cells. And if there is any doubt whether your cells are contaminated or not, you can check the same by DNA fingerprinting, karyotype analysis, etc. So, what about bacterial contamination? They are large, ubiquitous group of unicellular microorganisms. We all know that, and we can identify them from their shapes, ranging from spheres to rods and spirals. Because of their ubiquity, size, and fast growth rates, bacteria, along with yeast and molds, are most commonly encountered biological contaminants in cell culture. So, how do you know if your cell culture media is affected? So herein, I will introduce another very important component of your media that we discussed, but we didn't discuss the role. So in media, I have showed you a slide wherein you have seen the media was red in color. So why is the media red in color, right? What is the pigment in it? So the pigment in it is a pH indicator, which is phenol red, right? So phenol red is reddish at a neutral pH around 7 or 7.4. But once the media turns acidic, it becomes orangish or yellowish in color, in appearance. And if the media is contaminated with bacteria, then further the clarity of the media will disappear and it will start appearing cloudy, which will indicate the growth of microorganisms. So here is a flask, a cell culture flask, which is showing a yellowish media due to excessive growth of cells and not only animal cells, but also uh, contaminating microorganisms. and which is why the media is turbid and yellow in color, as indicated by the pH indicator phenol red. Okay, so this image here shows a phase contrast image of adherent 293 cells contaminated with E. coli. So once the contamination sets in, you cannot see the cells anymore, and your animal cells anymore, and it will soon be covered with bacterial cells. The other type of contamination is mold contamination, and molds are belonging to the kingdom fungi, and they grow as multicellular filaments called hyphae. We know that the filamentous form is called as the hyphae. Right. So similar to yeast contamination, the pH of culture remains stable in the initial stages of contamination, then rapidly increases as the culture becomes more heavily infected and becomes turbid. So what is the best way to identify yeast contamination? You see the wisp-like filaments or the threads, also called hyphae, and hyphae together form a bundle of mycelia, right? And which is the body of the yeast. And so that is the characteristic feature to identify mold contamination or yeast contamination. So this is how your yeast will seen or appear in cell culture as single-celled fungus. But in case of mold contamination, you can see more of these wisp-like filaments and clearly identify the contaminating uh, microorganism. So what in case of virus contamination? You cannot observe, obviously, viruses in light microscope because of their extremely small size. So what do you do instead? You can go for an electron microscopy, which is a relatively expensive technique, but you can also go for immunostaining with a panel of antibodies. You can go for ELISA assays, or you can do PCR-based diagnosis. Lastly, we come to mycoplasma contamination. These are simple bacteria that lack a cell wall, and they're considered the smallest self-replicating organism. Because of their extremely small size, mycoplasma are very difficult to detect until they achieve extremely high densities and cause the cell culture to deteriorate. Until then, there are no visible signs of infection. And from my own cell culture experience, I have seen this, and I stand by this statement that you cannot understand them or understand any mycoplasma contamination till previous night, and then come back to lab the next morning and find uh, no cells in them at all, no animal cells in them at all. Instead, small black dots appearing throughout the bulk of the liquid. Yeah. So finally, we will end our discussion with how you can detect mycoplasma infection of cell culture. You can you can either identify the black dots when the cells are already lost, or you can routinely check your cultures by fluorescent staining of nucleus, ELISA, PCR, immunostaining, autoradiography, microbiological assays, or electron microscopy. So in the next slide, I'll show you that if you use a nuclei-specific stain, here in this particular case, mycofluor mycoplasma detection kit has been used to stain the culture. And you can see here, while the animal's cells are showing clear, well-defined nucleus, circular in shape, big circles, the and there are no other dots coming from around the nucleus. But in second case, you can see that around the uh, big circles, there are small blue dots appearing near the big ones. So what that indicates is the presence of some contaminating microorganism which are having their own nuclear material, and the microfluor has stained them, and they are appearing as small blue dots. So that will indicate the presence of mycoplasma. So with that, we end our fourth lecture of the series. and there are more to come. Stay tuned.